Hi, I'm Jamie Norris, and this is my Neuromatch Flash Talk. The talk is titled Clustering and Forecasting of Interictal Epileptiform Discharges Using Machine Learning, and it's alongside my supervisors Carl Friston, Richard Roche, Gerald Curé, and Martin Tisdall. For a bit of context, epilepsy is drug resistant in around a third of cases, and for many of these people, surgery is a safe and effective option. For this work, I've used recordings from 2 pediatric patients that underwent pre-surgical evaluation at Great Ormond Street Hospital. And these recordings are with SEEG electrodes, so depth electrodes. The idea is that we want to try and learn more from the periods in between seizures, as these make up the majority of the time in recording, as well as being sort of less traumatic and less dangerous for the patient. And one such event that occurs during these periods are interictal epileptiform discharges, which are pathological hypersynchronous patterns of brain activity without observed clinical seizures. And firstly, their propagation can provide useful information for localizing the seizure focus. This isn't such a clear-cut relationship, but it helps motivate the first task that I'm going to talk through. And the second task I talk through is motivated by the fact that they can be considered markers of brain irritability. So for the first task, we've performed clustering of interictal epileptiform discharges based on their propagation through the brain to try and find different sort of populations of IEDs. And the first step was just to identify these actual spikes individually per channel. And then secondly, to group these up across channels and come up with sequences from them, depending on the order they occurred in, like this. Next, we produced a distance matrix for every pair of IED sequences, using the Levenshtein distance to try and sort of work out a distance that factors in the ordering of these IEDs sequences. And finally, this distance matrix is fed into a density-based clustering algorithm, dbScan, to get results. So here are the transitions for all of the IED sequences for patient 1. And if you look at just what came up in the first cluster, and there was only one cluster for this patient that was found, we can see transitions both within and between 3 separate electrodes. And it should be noted that these are not symmetrical about the diagonal, and quantifying this asymmetry can actually tell you the direction they more frequently occurred in. And by doing so, we can actually see along each side of the diagonal there's a sort of consistent color showing that there's a consistent direction that they travel in more frequently. And for example, one paper has shown that these tend to travel towards the seizure onset zone, so it could potentially be informative. For the second patient, there was a more widespread irritative zone, as you can see from the transition matrix, and 3 clusters were found for this patient. Here's the first one, which occurs over 2 electrodes generally. And again, we can quantify the degree of asymmetry to show a direction that they generally move towards. So the second task that we covered was motivated by single pulse electrical stimulation, which is an investigational tool in epilepsy surgery. And this can be used to probe epileptogenicity and seizure networks, with the idea being that you can sort of probe a pair of electrodes and measure the responses at other locations. And these responses can differ between different brain states. So for example, during non-REM sleep, it's been found that the high gamma activity component of these M1 responses were higher, and that can potentially make them more informative. There are also some other response differences and some stochastic elements that potentially rely on the brain states. So the idea is that we want to be able to forecast IEDs using these as a proxy for the brain irritability to help guide SPES. So for example, when more IEDs are forecasted to occur, it might be a better time to perform SPES. So we formulated this as a pseudo-prospective task where the majority of the data was used for training purposes. An hour of data was then left out, which would theoretically be ignored in real time, where you would do the model training and then evaluation took place in the remaining hour of data, which is when in theory you'd apply SPES. These models were patient specific because the networks did sort of vary a lot from patient to patient, and the recording locations also do differ a lot. And these regression models, but during post-processing, so at inference time, you would in theory use a threshold to decide whether or not to apply SPES. So we use patient-specific thresholds here, reformulated as a classification task. And the steps involved in training were to first segment the data into second long segments, then to apply pre-processing and feature extraction. And the features that we used were spectral power, phase amplitude coupling, as well as some summary statistics of the correlation matrix between the different channels and the number of previous IEDs that occurred, in this case in the previous minute. And then these were each labeled with the number of IEDs that occurred in the following second. Once this data set was created, supervised learning took place to produce the regression models, and gradient boosted decision trees were used for this, which are a form of decision tree ensemble. The results did differ between the two patients. So as you can see here, for patient 1, the best sort of feature subset gave a ROC of around 0.8, whereas for patient 2, it was around 0.6. And by doing some further testing, it could be seen that, so we'd previously seen, in fact, that there were sort of the IED counts did vary over time. So there were some cycles, and depending on which part of the cycle your test data ended up in, this seemed to have an effect on the actual score itself. So by instead training on one cycle, for example, and evaluating on a separate cycle for each of the two patients, a more similar score was found. So it was 0.75 versus 0.65 when we just did that instead. And of course, that doesn't make the task studio perspective, but it gives you an idea that there was a reliance on the actual test set itself, as the performance does change over time a fair bit. So the next steps are going to be, firstly, to test if more predicted IEDs actually did correspond to more informative SPECT responses, to improve the IED detection algorithm, which is so far just a threshold-based detector, to expand to a larger cohort, as only 2 patients have been used so far. And finally, to use dynamic causal modeling to link to the underlying biology. So to see what are the actual differences between the periods where there's expected to be less IEDs versus periods where there's expected to be more IEDs. And thank you very much for listening. And thanks again to my supervisors, as well as Ashwin Chari, for helping with the data.